Okay, data safety. So, first of all, why are we talking about data safety in a Python class? Well, it's common for folks to learn Python in preparation for projects or careers in data engineering or data science or machine learning, and all of those roles require you to be able to interpret data and draw conclusions from it. But here's the danger. There are a lot of ways to generate numbers, and one number doesn't necessarily look any more suspicious than another number, regardless of how that number was reached. This is why it's so easy for research organizations with different slants, for example, to draw two different conclusions from the same data. But what we're going to talk about today is not intentional slant. Instead, we're going to talk about some honest but costly mistakes that often happen in bona fide scientific research papers published in hard science journals. The researchers didn't necessarily intentionally slant the paper, or leave out data, or present something as more sure than it really was, but they just didn't have the statistical safeguards in place to come to a sound and accurate conclusion about the things they were researching. I'm teaching you statistical safety in a Python class for the same reason you'd learn knife safety in a knife skills class. You can impact lots of lives with the work you're going to be able to do as a Pythonista. And if it's data manipulation or interpretation work, I want to make sure that you have some idea of what's going on. So, let's talk about how we use data. We use data to build models. Now, in the literal sense, we use them to build machine learning models, but we also use them in a more general sense to shape our mental models, our understandings of cause and effect. Models are almost never 100% accurate. Rather, they're a simplified representation of a phenomenon, and that gives us a way to wrap our heads around the phenomenon. So, when you analyze a set of numbers, you have to keep in mind that that set doesn't represent all the numbers in the world on this topic. Take, for example, your height. How tall are you? Now, could you use that information to extrapolate that everybody in the world is the same height as you? Of course not. It's just one data point. Now, imagine that instead we took the heights of everyone in this class. Would that give us a better idea of how tall people are in the world in general than just your height? Now, it would, but it still wouldn't be the full picture. We probably don't have the tallest or the shortest person in the world in this class. The average height in this class might not really match the average height of everyone in the world, especially because huge parts of the world's population are completely not represented in this class. Most data analysis attempts to draw conclusions about something based on an incomplete subset of the total data about that thing. Statistical methods give us a way to quantify how wrong we might be about our extrapolations. That's where it's helpful to use a distribution and standard deviation. Let's talk a little bit about what that means now. I know I already have some statisticians in the room, so I'll keep this quick. Basically, when you take samples of data, that data can be distributed in different ways. Here are some examples. Maybe your data skews towards bigger numbers. Maybe it skews towards smaller numbers. Maybe it's all over the place. That having been said, in a lot of cases, the data tends to be around a central value, like this, and the other numbers kind of fan out from that. So in the case of heights, for example, probably this large part in the middle of the graph of all the heights in the world happens somewhere between 5 feet and 6 feet high, right? Now there's people taller than that, and there's people shorter than that, but it tends to be most common to be between 5 and 6 feet high. So we're likely to see a curve like this on people's heights. It's called a bell curve because it looks kind of like a bell. The scientific term that we typically use for it is the normal distribution. The way that a semi-random variable is normally distributed over a large population of subjects. So that's the normal distribution. Now, we'll talk a little bit more about distributions later, but you can think of a distribution as the way that a set of data might be allocated, both in your data set and in the real larger world that your data set is attempting to model. Now, a standard deviation is a characteristic of that distribution. It represents how spread out the numbers are. Now, let's talk about this example again in terms of height. We have a certain distribution of heights among humans, right? Most people probably fall between 5 and 6 feet high, and the vast majority of people probably fall between 4 and 7 feet high, and maybe all people fall somewhere between 6 inches and 12 feet high, right? So we have this distribution, whereas there's this spread of a relatively small amount, let's say 24 to 36 inches, maybe about a meter, in the middle that most people are kind of within. So those numbers aren't particularly spread out. Suppose, though, that we were instead taking the heights of all of the horses in the world. Now, there are miniature horses who are 12 or 16 inches tall. And then there are, on the other end, massive draft horses who can be 6 or even 7 feet tall at the shoulder. Now, it might be that humans sort of like minimum and maximum are similar to horses. And so we're talking about a distribution of roughly the same range, but it's extremely uncommon to have a human who's above 7 feet tall. Whereas with horses, there are particular breeds that are almost always that tall or taller. And similarly, it's relatively rare for people to be 16 inches high, but there are whole types of horses, miniature horses, for example, who are regularly approximately that height. So even though we have similar minima and maxima on human heights and horse heights, the standard deviation on the horse heights is going to be wider than the standard deviation for human heights. It's going to be larger than the human height standard deviation because the numbers are far more spread out over the range than human heights are. Now, we can use the standard deviation as a measure of where the population probably cuts off and where most of the population is. And that's why when since humans have a smaller standard deviation on their height numbers, the vast majority of people are going to be within a smaller range of heights, whereas with horses it'll be much larger. Now, what does any of this have to do with the statistics that we're talking about? So, we need the standard deviation to calculate a confidence interval, and we'll use a distribution to do that too. Now, here's the question, right? What is a confidence interval, and what does it help us figure out? Why do we need this? Well, a confidence interval helps us answer the question based on how much data we have sampled and how much the values in that sample differ from each other. How far away from our sample mean could the true mean be? For example, suppose that I have 200 different horses and their heights listed in my data set. Based on the fact that I sampled 200 horses, and based on the mean of, let's say, 5 feet high, and based on how spread out the numbers are in my 200 samples, whatever the mean is that I got, let's say 5'4" at the shoulder, 16 hands, if that's my mean, where is the range of numbers where it's 95% certain that the true mean, the mean of all horse heights in the world, is within this range with 95% confidence? So, in the words of Simply Psychology, the 95% confidence interval is a range of values that you can be 95% certain contains the true mean of the population, of all the population that your data represents. Now, as the sample size increases, the range of interval values will narrow, meaning that you know that mean with much more accuracy compared to a smaller sample. So, if I have 200 horses in my data set, my confidence interval is going to be somewhere around my mean. My error bars around where the mean of all horse heights could be is going to be much larger, versus if I have 100,000 horses in my data set and I get an average height of 100,000 horses, that is much more likely to be closer to the true mean than a mean with only 200 horses. And so my confidence interval for that much data is much smaller, because I can be much more sure with 95% accuracy, for example, that my true mean of all horses falls somewhere close to the mean in my data. So we can look at some visual representations of this as well. So it might be that your data is super spread out or super narrow, which will result in a wider or less wide confidence interval. This first diagram might represent, for example, human heights. And those human heights are not that spread out, and so we have a narrower bell and a narrower confidence interval. This second one, however, might represent horse heights. We've got a wider confidence interval there because we have a larger standard deviation, more distribution among the heights. Now, we could also get a larger and smaller confidence interval based on amount of data. In this third image, what we see is what we might expect if we took a bunch of heights and we had 100,000 data points. Enough data points that if this was our mean among all of our subjects, we're certain that the true mean is somewhere in here. Whereas if I only have 200 subjects, even with the same mean, I have less certainty around where that mean is, and so I have to have a much wider range of numbers that might be the true mean. So typically, we use the normal distribution for calculating confidence intervals. And that's because the normal distribution tends to give us a good approximation when we're talking about a normally distributed variable with more than 120 samples. Now, it's also the case that you'll often be asked to look at numbers for which we have fewer than 120 samples. For example, maybe only 30 samples of a water reading, or 60 samples of a course review by several students. Now, for those relatively small numbers, we can use a different distribution. We call it the t-distribution, or the student t-distribution. It's wider and flatter than the normal distribution up until about 120 samples, when it starts to look more like the normal distribution. That gives us a better picture of our confidence interval for those smaller numbers, because the wider and flatter nature of it makes it more likely that we're going to get the appropriate interval for a small sample size. Now, I'm not going to have you write code that calculates the confidence interval. I'm less concerned with you knowing the exact details of the implementation of the confidence interval than I am concerned that you understand what it is and when you need to use it during data analysis. So, I've written some code for you that will allow you to get confidence intervals of numbers in data frames when you're doing data analysis. That method calculates confidence intervals based on the mean, the size, and the standard deviation of several sets of data. It has a default confidence interval of 95%, but you can change it if you want to. There's a default on the variable, but you can pass in a different one. What it returns is the upper and lower bounds of the confidence interval around the means that you passed in. Now, precisely that means that this function answers the question. Given this mean, standard deviation, and number of data points, there is a confidence probability, that is the number that you passed in our case defaulting to 0.95, that the true mean for all of the data that this data represents is between what lower number and what higher number. Okay? Now, it's worth noting that this function takes in collections of data summaries, not just individual data summaries. So you could pass it in a list of sample sizes, numbers of items in collections, a list of standard deviations representing those collections, and a list of means for each of those collections. And it's going to calculate your confidence interval for each of those collections above and below in sequence. That means that this method is a collection method. It takes in collections of values and it does an operation on each one. It's useful for calculating confidence intervals for every mean, for example, and a collection of means that you stored in a data frame. So you can pass series into this method and get out all of the confidence intervals for a given column. You can also use this function on just one set of summary data, but you have to pass in the sample size, standard deviation, and mean of that data set inside of brackets to make a one-item collection, like this. Now, let's see the method in action by calculating confidence intervals around the number of days that various Chicago municipal maintenance operations took. So this is a data set that lists several different things that had to be done for maintenance in Chicago, the target number of days it was supposed to take, and how many days it actually took. Several readings for every different year, right? So we have an average number of days that it took, and we have a target number of days that it took. So we can subtract the target number of days from the average number of days to get the slippage, how late each of these tasks ended up being turned in on average in the city of Chicago. Now what we're going to do is we are going to calculate the confidence interval around that average slippage based on how many data points we had for each year. And the place where we do that is right here. So I'm going to make a new column in my data frame for the bottom of the confidence interval and the top of the confidence interval by calling confidence interval for collection. Oh my goodness. By calling confidence interval for collection on all of my activity counts, all of my standard deviations, and all of my means. And when we do that, we see that we have the average days to complete the activity, mean, and then the average days to complete the activity, standard deviation, the count, in this case 3, the bottom of the confidence interval and the top of the confidence interval. Now, one thing you're going to notice here is that this confidence interval is really big, right? We've got a confidence interval of, it looks like 68 or something, around this mean of, larger than 68, somewhere around 70 something, around this mean of 37. And the reason for that is that the activity count is so small that the mean of these 3 activities barely represents what the mean really could be. Now, if we scroll down, we'll see that the confidence intervals get much smaller when we're talking about larger data sets. Now, it's a little bit tough to tell in this specific example because the number of days that things were supposed to take is so different from these bottom ones versus the ones on top. So here we have a target response days max of 180, and down here we're expecting just 1. But it's worth noting that you are going to end up with a smaller set of error bars around your mean if you have taken that mean from more data points. So that's confidence intervals. Now let's talk about t-tests and what they're useful for. A t-test helps us answer the question, is the distance between 2 means statistically significant? Suppose I have 2 separate populations and their means are different. How do I know if that explains a change or if it's just random? So it's possible that those 2 different means represent a statistically significant difference even if their confidence intervals overlap. For that, we could take a t-test. Once again, I'm not too concerned about you understanding the implementation of a t-test in this lecture as much as I want to make sure that you know what it is and when to use it. So again, I've written you code. This function performs a t-test for 2 data sets, given their sizes, their standard deviations, and their means. It has a default confidence of 95%, but as before, you can change it. It returns a tuple of whether to accept the null hypothesis and then the t-value. Now, what do I mean, accept the null hypothesis? So, in statistics, you'll often hear about rejecting the null hypothesis, which means that we think there's a difference in this data. The null hypothesis is the hypothesis that your data's differences are due to random chance and don't necessarily represent a true difference in the overall populations, with a difference maybe in your measurements of those populations. That's the null hypothesis. To reject the null hypothesis is to say, I don't think random chance explains why these two numbers are different. I think that there is some causality at play here. So when we accept the null hypothesis, what we're saying is the difference between these two means is not statistically significant. Or more precisely, there is some probability, 1 minus the confidence, in this case 0.95. So there is a 5% probability that the difference between these two means represents an actual difference between these two data sets. And customarily, if there's a less than 5% chance that the difference represents some actual significant finding in the data, then we say we have not found something statistically significant. Now that 95% 5% number does depend a lot on what field you're in, what kind of analysis you're doing, how much data you've collected, and so on and so forth. Well, we're continuing to use it here because it's the one that you're going to see the most often in statistical tests. So what you're going to do here is you are going to pass in, what you're going to do is you will pass in the number of samples of your first collection, the standard deviation of your first collection, and the mean of your first collection. And then the same thing for your second collection. And what it's going to do is it's going to give you whether you should accept the null hypothesis and your t-value. And so you can see that down here. Where are we? Yeah. So return, accept the null hypothesis, and the t-value. Okay? So if, for example, we wanted to do a t-test for two different means, and let's say that I've got, what are we talking about? Number of samples, let's say 200,000 samples, right? So we got a big data set at the beginning. And then let's do a standard deviation of 0.05. Where is my code? 0.05 and a mean of 0.62. And then we're going to do another collection, 100,000 data points, standard deviation of 0.01 and 0.95. So what it tells me is I should not accept the null hypothesis. It seems like these two data sets do have statistically significantly different means. Now, suppose, for example, that my two standard deviations were the standard deviation were bigger, or these means were a lot closer together. And let's suppose that I also had a lot less data. Let's make it real small. Now what I'm going to end up here with is true. And in this case, what we're saying is you should accept the null hypothesis for this data. It is likely that the difference between these two means could be explained by random chance, specifically 95% likely or higher. That the difference between these two means could be explained by random chance. Now, we've talked about confidence intervals and we've talked about t-tests. A lot of the time, business people are going to make decisions based on averages and nothing else. They're not going to do a t-test on two different averages. They're just going to look at it with their eyeballs, determine that the two averages are different, and say, at face value, these two things must be different because some external factor is explaining it. Or they're not going to run confidence intervals around their averages. You're going to see this all the time, taking averages at face value. Now, for business decisions, it's often fine, and it's not necessarily worth the effort to really be statistically rigorous on every business decision. However, there are situations where you need to be using the confidence interval, like weight tolerances for the materials being used to build a bridge. temperature tolerances for the O-rings on the solid rocket boosters for a space shuttle, the amount of a toxic substance in a municipal water source, or the effectiveness of a vaccine, just as examples. Any situation where people's health, safety, and quality of life depend on how you act on this data merits a confidence interval. So that's the math part. Now let's talk about some plain old study design problems that you'll want to watch out for if somebody asks you to analyze a bunch of data. The first one is extrapolating based on a non-representative subset. A confounding variable is a factor that affects data that you don't account for in your analysis that impacts your results. There's an effect in statistics called Simpson's paradox that sounds very fancy, but it ends up being basically this. The effect happens when subgroups of data show one trend, but the entirety of the data shows the opposite trend. Let's talk about an example. A 1973 study of gender bias at UC Berkeley revealed that, on the whole, women were being admitted to the school at a lower rate than men. It turned out, though, that when the data was broken down by department, women were admitted at a higher rate than men. Here's what happened. Women were applying to the programs that were harder to get into, more selective. It's really important to track down your confounding variables as much as possible so you don't accidentally make a claim that misinterprets its causality. Here's a common medical example that's been an issue for ages: body mass index. Body mass index scores are used to determine how healthy people are, supposedly, based on their weight to height ratio. The problem is that the BMI scale was developed based on a sample of white people and overrepresents white men in particular. It does not extrapolate well to other people's bodies, and it especially pathologizes black women's bodies. As a result, doctors deny care to people who need it because those people need to lose weight first, according to a metric that was not built for their bodies. One way to track down confounding variables is to make sure that the distribution of your sample set on each variable you have on record matches the distribution of the population you're trying to extrapolate to. Doctors right now, for example, are trying to ensure that the distribution of their study participants for COVID vaccine trials matches the distribution of people in the broader world who will get the vaccine. That is, in particular, they want the trials to include people of all ethnicities, so they don't make a safety claim based only on white people and then start injecting a substance into people's arms with no information about how it might affect them. Getting study participants has been difficult because this country's history with performing reckless medical experiments on black people in particular, for example, in this Tuskegee study, gives black and indigenous people of color really good reasons to be wary of the medical establishment. So these are the kinds of issues that do affect our science in real life and end up having implications for policy and implementation decisions that affect us every day. Let's talk about another study design issue to be aware of: multiple comparisons. You may have 95% certainty that any one comparison isn't statistically significant by fluke. But when you run a bunch of comparisons, eventually one of them is going to be a fluke. In fact, when you run 100 separate comparisons, your likelihood that none of the significant outcomes are flukes drops to a measly 1%. You're almost certain at some point to detect a statistically significant difference that is due to random chance. Because you have that 95% confidence interval, right? There's still that 5% chance that these two numbers being different is a result of a fluke. Now, there's a comic about jelly beans that illustrates this really well, and I want to show it to you right now. So here's what happens. Let me see if I can make this a little bit bigger for you. There we go. So there's this claim that jelly beans cause acne. Okay? And scientists are asked to investigate. So they investigate all the jelly beans and they don't find a difference. So then those scientists are asked to check every individual color of jelly beans separately. And they start to check. They check purple and brown and pink and blue and teal and salmon and red and turquoise, magenta, yellow, gray, tan, cyan, green, and yellow. All of them are showing no statistically significant difference except for green, which happens to show a statistically significant difference by chance. They do all these different colors. And you can imagine if you checked every single flavor of the Jelly Belly brand of jelly beans for being correlated with acne, by random chance, you might end up with one that does. And that's often what happens, and we get news like this, green jelly beans linked to acne, even though what actually happened there is we just made a whole bunch of comparisons until we found one that had a correlate by pure random chance. By the way, this is one of the reasons that most scientific studies that say blah, blah, blah food helps you live longer are mostly garbage. Is because what they did was they took a whole bunch of people who lived various lifespans and they looked at everything those people ate, and one of them may be correlated by chance. So that's what's happening there. Now, we want to make sure that we are not doing that in our studies, just making a whole load of comparisons and then finding something that randomly correlates with our target variable and claiming that that's the cause. So there is one solution for this. If, for example, we're analyzing a bunch of data, we're trying to find a cause, and we want to analyze a whole bunch of different columns for possible correlation. That solution is called the Bonferroni correction, and it basically is to divide your intended p-value by the number of comparisons you're running. So if you've got a 5% p-value, you want to make sure that there's less than a 5% chance that the difference is due to random chance, and you want to check 3 variables, then you're really looking for a p-value of 5 divided by 3, or like 1.6 something p-value for each of those, right? Now, this method gets some criticism for being too harsh and potentially missing important findings, because when you divide a p-value of 0.05 by 3, you're now talking about a really small number. So for that reason, sometimes folks temper it by lowering the p-value if they're making multiple comparisons, but not necessarily all the way to the Bonferroni correction prescription. Now, I don't want you to worry too much about how much should I correct for multiple comparisons. First, I want you to be aware of when you're making multiple comparisons and identify that in those cases, a result that would be statistically significant in the case of one comparison might not necessarily be statistically significant for you. Now, the final mistake that I want to talk about is an analysis error, and it's called the continuity error. So, this type of data representation error happens when we misinterpret continuous data that doesn't fall into discrete categories like body mass index, and either misrepresents it as discrete categories or interprets it in some way that isn't true to the data. And I think an example is really helpful here. Once again, I got to pick on body mass index. So, with BMI, we frequently see 2 categories, normal weight and overweight. So, as we've already discussed, body mass index as a metric in the first place has been demonstrated to be a poor measure of health and fitness. Already talked about that issue. Let's stick to continuity errors specifically. Frequently, BMI data gets categorized as normal weight being 24.9 or lower, or overweight above 24.9. Where is underweight? Also, what is the difference between a 24.8 and a 25.1? When these middle values get averaged together with extremes on either end, it looks like this 0.2 BMI difference in the middle is a night and day difference. It's not. We're just representing a wide range with a tiny number of categories, and lumping in folks who have a 25.1 body mass index as having identical bodies to people with a 32, 34, or 40. And lumping in people with a 24.9 body mass index as having identical bodies to people who have a BMI of 20 or 18 or 16. What we have is a continuous variable, and what we've done is we've divided it artificially into 2 categories. Now, it's worth examining whether and why we need to categorize continuous variables before we do it. There are good reasons to do it, like get evenly sized buckets of points to compare means, or draw meaningful visualizations, etc. So maybe if we were looking at height data again, we don't want to take each individual data point on its own, but we want to make buckets. This many people are between 5 feet high and 5'5". This many people are between 5'5" and 6 feet tall. this many people are between 6 feet tall and 6'5", and so on and so forth, right? So it can be useful, but it's not a default thing to do. And it's something that it's very important to be aware that you're doing when you do it, so that you don't accidentally draw conclusions based on a categorized representation of continuous data.